Group number 3, can you please start your presentation?
Receive group, please.
Ma'am, can I start, ma'am?
Yeah. You see my screen now?
Yes, ma'am.
Okay, please.
A very pleasant afternoon to my friends. Myself, Shivani Krishnan, and my friends are here to present about autosomal dominant and autosomal recessive. And I'm here to give an intro about autosomal dominant and recessive. Autosomal dominant. Autosomal dominant is a pattern of inheritance characteristic of some genetic disorders. Autosomal means that the gene in question is located on one of the numbered or non-sex chromosome. Dominant means that a single copy of the mutated gene, that is from one parent, is enough to cause the disorder. A child of a person affected by an autosomal dominant condition has a 50% chance of being affected by that condition via inheritance of a dominant allele. By contrast, an autosomal recessive disorder requires two copies of the mutated gene, that is from one from each parent, to cause the disorder. Huntington's disease is an example of autosomal dominant genetic disorder. And this is the autosomal dominant example, a picture of example. It is a pedigree chart of autosomal dominant. Next, autosomal recessive. Autosomal recessive is a pattern of inheritance characteristic of some genetic disorder. Same thing, autosomal means that the gene in a question is located on one of the numbered or non-sex chromosome. Recessive means that the two copies of the mutated gene, that is from one from each parent, are required to cause the disorder. In a family where both parents are carriers and do not have the disease, roughly a quarter of their children will inherit two disease causing alleles and have the disease. By contrast, an autosomal dominant disorder requires only a single copy of the mutated gene from one parent to cause the disorder. Sickle cell anemia is an example of autosomal recessive genetic disorder. Next slide. This is the autosomal recessive pedigree chart, and I give a short clue about autosomal dominant and recessive. That's a common point. All affected offsprings has affected parents. This is the common point for autosomal dominant and recessive. If all affected offspring has affected parents, if that is yes means then it is dominant. If that is no means it is recessive. A very important clue for autosomal dominant is all affected males have affected mothers. All affected males have affected mothers, and all daughters of the affected males are also affected. This is the clue for autosomal dominant. Mother to son, son to daughter. You have this in your mind, like mother to son, son to daughter. Two females and one male, which means mother to son and son to daughter. For recessive, a clue, all the affected females have affected fathers. All affected females have affected fathers, and all her sons are also affected. It is opposite like of dominant. The recessive is opposite like dominant. All the affected females have affected fathers, and all her sons are also affected, which means the female will be common here, and the female will give father will be affected. That means male will be affected, and her sons will also be affected. Here the female is common, the fathers will be affected and the sons will be affected. This is the very important and very useful clue for the autosomal dominant and recessive. Thank you. Now the genetic risk will be explained by Kidara.
Okay, thank you. Otana.
Good afternoon, everyone. Now
Yeah.
I'm going to explain about the genetic risks. There are two types of genetic risk. They are autosomal dominant genetic risk and autosomal recessive genetic risk. Firstly, let's see about the autosomal dominant genetic risk. Each gamete from an individual with a dominant trait or condition will contain either the normal allele or the variant allele. If we represent the dominant variant allele as capital D and the normal allele as small d, then the possible combinations of the gamete is seen in any child born to a person affected with a dominant condition has a 1 in 2, which means 50% chances of inheriting it and being similarly affected. And then autosomal recessive genetic risk is if we represent the normal dominant allele as capital R and the recessive variant allele as small r, then each parental gamete carries either the variant or the normal allele. At conception, the various possible combination of gametes means that the offspring of two heterozygous have a 1 in 4, that is 25% chances of being homozygous affected. 1 in 2, 50% of chances of being heterozygous unaffected, and 1 in 4, that is 25% chances of being homozygous unaffected.
Now some examples will be explained by Abhinaya Jyoti.
Let's see the examples of autosomal dominant Marfan syndrome, Huntington disease. Let's see the example of autosomal recessive cystic fibrosis, sickle cell anemia, Tay-Sachs.
Now the Marfan syndrome will be explained by Sham.
I'm going to explain about Marfan syndrome. Marfan syndrome is a genetic condition that affects connective tissue, which provides support for the body and organs. Marfan syndrome can damage the blood vessels, heart, eyes, skin, lungs, and the bones of the hips, spine, feet, and ribcage. And its causes. Marfan syndrome is very rare, happening in about 1 in 5,000 people. Marfan syndrome is caused by a mutation in a gene called FBN1. The mutation limits the body ability to make proteins needed to build the connective tissue. 1 in 4 people with Marfan syndrome develop the condition for unknown reasons. A person with Marfan syndrome has a 1 in 2 chance of passing it on their children. And its symptoms: arms, legs, fingers, and toes that are longer than expected, taller, taller stature than expected for the family, breastbone that sticks out or in, crowded teeth, curved spine, flat feet. Thank you.
Now the Huntington's disease will be explained by Harish.
Good afternoon, ma'am. Myself Harish. I'm here to explain about Huntington disease. Huntington disease is a progressive brain disorder caused by defective gene. This disease causes changes in central brain of the brain and affects movement, mood, thinking skills. Huntington disease is caused by single defective gene on chromosome 4 on the 23rd human chromosome that carries person's entire genetic code. Symptoms: Huntington disease usually develops between age 30 to age 50, but can appear as early as age 2 and late as 80. The hallmark symptoms of Huntington disease is uncontrolled movement of arms, legs, head, face, and upper body. Huntington disease also causes a declining in thinking and reasoning skill, including memory, concentration, judgment. Thank you.
Now, the pleiotropy will be explained by Rishikesh.
Pleiotropism. Prion means more and troops means ways. When a single pair of gene controls the production of many characters, then it is called pleiotropism. The gene is called pleiotropic gene, and the term is coined in 1910 by
And
okay. So we will see examples of pleiotropism, which is phenylketonuria. This disorder is caused by a deficiency of the enzyme phenylalanine hydroxylase, which is necessary to convert the essential amino acid phenylalanine to tyrosine. A defect in a single gene that codes for this enzyme therefore results in the multiple phenotypes associated with the PKU, including mental retardation, eczema, and pigmentation defects that make affected individual light-skinned. Understand?
now, the autosomal dominant of variable expressivity to new variants and reduced penetrance will be explained by Priyadarshini.
Variable expressivity. In genetics, the expressivity is the degree to which a phenotype is expressed by an individual having a particular genotype. That is, variable expressivity describes when one genotype produces a range of phenotypes. Alternately, it may refer to the expression of particular gene by individual having a certain phenotype. It is also expressivity is related to the intensity of a given phenotype. It differs from the penetrance, which refers to the proportion of individuals with a particular genotype that actually expresses the phenotype. It variable expressivity. It also means that one gene can affect one individual much more severely than another individual. Penetrance here refers to the proportion of individuals whose phenotype matches their genotype. Reduced penetrance, a term that describes when some people with a disease causing mutation, that is change in a gene, develop the disease while others don't. For example, certain gene mutations causes cancer, but because of reduced penetrance, not everyone who has the mutation will develop cancer. But currently, there is no way to know which people who have a cancer causing gene mutation will develop cancer, also called incomplete penetrance. Incomplete penetrance is used to when discrete categories are defined. New variant, the sudden, unexpected appearance of a genetic condition arising as a result of a pathogenic heterogeneous gene variant is called de novo variant. The astute clinician also needs to be aware that the family relationship may not be as stated, that is, there may be undislocalized non paternity or rarely non maternity. Thank you.
Now, the codominance will be explained by Abhinay Jyoti.
Codominance. Codominance is the term used for allelic traits, both expressed in heterozygous state. For example, person with blood group A and B. It is possible to demonstrate A and B blood group on the red blood cells. So the A and B blood group are example of codominant. Thank you.
Now, the homozygosity and consanguinity will be explained by Anjana.
Mostly, autosomal dominant condition only occurs in heterozygous state. However, occasionally, children who have both parents as heterozygous for dominantly inherited disorder are in risk of being homozygous for gene variants. For example, we can take achondroplasia, which is the genetic condition that prevents the protein in the body of growth. And the next example is hypercholesterolemia, which is the excess cholesterol in blood. And now we can look at consanguinity. Consanguinity refers to relationship by blood or a common ancestor. Inquiry into the family history of individuals affected with rare recessive conditions might reveal that their parents are related, which is aka consanguineous. The rarer a recessive trait or disorder, the greater the frequency of consanguinity among the parents of affected individuals. For example, we can take cystic fibrosis, which is most common in Western Europe, and where the frequency of parental consanguinity is only slightly greater than the common population of the world. By contrast, when Bateson and Garrett originally described the very rare alkaptonuria, they described that a quarter or more of the parents were first cousins, and rightly reason that rare alleles are more likely to meet up in the offsprings of cousins than unrelated parents. In large inbred kindreds, an autosomal recessive condition may be present in more than one branch of the family. Thank you.
And now let's see about the autosomal recessive disorders. Now the cystic fibrosis will be explained by Farheen Taj.
Cystic fibrosis is a fatal inherited disease found in humans and characterized by thick and sticky mucus, particularly in the respiratory and digestive system. Autosomal recessive meaning that is not inherited solely from mother or father as in sex-linked disorder. Rather, cystic fibrosis is inherited when an individual receives a mutated copy of a gene associated with cystic fibrosis from both parents. Diagnosis of cystic fibrosis. Adult and children can be tested to see if they carry the mutated CF gene. Genetic carrier analyzes a sample of DNA from each parent via a mouth swab or blood sample examined for mutations in the CF gene. Carrier testing is highly accurate, but some mutations are undetectable. Therefore, the person who tests negative for cystic fibrosis mutations can be as a carrier. Symptoms: persistent coughing at time with phlegm, poor growth or weight gain in spite of a good appetite.
Next, sickle cell anemia will be explained by Rishikesh.
Sir, can you change the slide? Next one.
Next one.
Next slide.
Yes.
Okay.
Yeah, this one.
Okay.
Okay. Sickle cell anemia. Other names of sickle cell anemia are HbS disease, hemoglobin S disease, sickling disorder due to hemoglobin S. Okay, now we see the summary. Sickle cell anemia is a disease in which the body produces red blood cells that are shaped like crescents or sickles. These cells do not last as long as normal or round red blood cells, which leads to anemia. Low number of red blood cells with anemia. Okay. The sickle-shaped red blood cells also get stuck in blood vessels, blocking the blood flow. And signs and symptoms of the sickle cell disease usually begin in early childhood and may include anemia, repeated infections, and periodic episodes of pain called a crisis. This condition is caused by genetic changes in the HBB gene and is inherited in an autosomal recessive pattern. Okay, sir, can you change to the previous slide?
Okay.
Okay. Okay.
Previous slide, ma'am.
Previous.
This one or this one? No, wait.
Previous.
This one?
Yeah. Okay. So the introduction to the inheritance of sickle cell anemia. Okay, everyone has 2 copies of hemoglobin gene in every cell in their body, apart from eggs and sperms. They get one from their mother and one from their father. When eggs and sperms are made, only one of the 2 gene goes into each egg or sperm cell. This is so that when the egg and sperm comes together to make a new baby, this new person that has 2 genes in every cell in their body as well, the gene the baby gets will therefore depends on the gene carried in its parents. Sickle cell anemia is called recessive condition because you must have 2 copies of the single hemoglobin gene to have this disorder. Sickle hemoglobin is often shortened to S or HbS. If you have only 1 copy of sickle hemoglobin along with 1 copy of more usual hemoglobin A or HbA, you are said to have sickle cell trait. This is not an illness, but means that you carry the gene and can pass it to your children. If you also have a sickle cell trait or sickle cell anemia, your children could get sickle cell anemia. As you can see in this picture, the when you have type of alleles, a pair of alleles, you are normal. And if you have AS and your carrier, you don't get the disease, but you will carry that disease to the next generation. If you're having SS, then it means you will have the disease. That's it.
Now, the Tay-Sachs disease will be explained by Jaypriya.
Tay-Sachs disease. Tay-Sachs disease is a genetic disorder that is passed from a parents to their children. It occurs when a child inherits a flaw in an extra gene from both parents. It is inherited by. This condition is inherited in an autosomal recessive pattern, which means both copies of a gene in each cell have a variant. The parent of an individual with an autosomal recessive condition each carries one copy of an altered gene, but they do not show the signs and symptoms of their condition. The next slide.
Okay.
Symptoms are being overly startled by a noise and movement, being very slow to reach the milestones like learning to crawl, losing skills that they have already learned, floppiness and weakness, which keeps getting worse until they are unable to move, difficulty swallowing, loss of vision or hearing, muscle stiffness, and seizures. So, next slide. Diagnosis. The blood test checks the level of exosaminide A enzyme in the blood. The levels are low or absent in Tay-Sachs disease. Genetic testing. These tests can examine the EXA gene to identify whether there are changes there that indicate the Tay-Sachs disease. Summary. Tay-Sachs disease is a rarely inherited condition that mainly affects the babies and young children. It stops the nerves working properly and usually fatal. Thank you.
Now, the pseudodominance will be explained by Yashwar. Next slide, ma'am.
Okay.
Pseudodominance. If an individual who is homozygous for an autosomal recessive condition has a children with a carrier of same condition, their offspring have a 50% chance of being affected. Such pedigree is said to be exhibited pseudodominance. A pedigree with a woman homozygous for an autosomal recessive disorder whose husband is heterozygous. For the same disorder, they have a homozygous affected doctor, so that pedigree shows pseudodominant inheritance.
Now, the locus heterogeneity and variant heterogeneity will be explained by Shrutika.
Locus
Hey.
heterogeneity.
Hey, sorry,
Locus
sorry.
heterogeneity occurs when multiple genomic loci are capable of producing the same phenotype, and each individual mutation is sufficient to cause the specific phenotype independently. For example, locus heterogeneity is the disorder retinitis pigmentosa, a retinal dystrophy resulting from the loss of photoreceptors in the retina, for which more than 45 genes have been identified. Variant heterogeneity. Heterogeneity can also occur at allelic level in the majority of single gene disorders. For example, beta thalassemia, a large number of different variants have been identified as being responsible. There are individuals who have two different variants at the same locus and are known as compound heterozygotes, constituting what is known as allelic heterogeneity. Most individuals affected with an autosomal recessive condition are compound heterozygotes rather than true homozygotes, unless their parents are related, in which case they are likely to be homozygous for the same variant by descent, that is, inherited from a common ancestors. Thank you.
Our PPT slides were prepared by Parthiban and Nihal. Thank you so much. Thank you.
Thank you so much for group 3. You have great presentation. Do you have any question for them? Yeah, you keep silent. That means you have no question. And so now next, we continue with group 2, right? Okay, please wait for me. I'm trying to do something. Can you see my screen now? Hey?
Yeah, ma'am, yeah, we can.
Okay. Yeah, okay. Group 2, okay, introduction of X-linked inheritance. Please.
Can we start, ma'am? Can we start, ma'am?
Yeah, you can start. Okay, please.
Good afternoon, everyone. A warm welcome to our respectable teacher and my dear friends who have joined this online presentation. We, the students of Group 2, are pleased to present the presentation on the topic X-linked inheritance. In this PowerPoint presentation, we would learn X-linked inheritance, dominant and recessive, genetic risks, and effects of X-linked inheritance. Now, I would like to call upon Vaishnavi to explain about X-linked inheritance.
Good afternoon, ma'am, and good afternoon, guys. I'm going to explain about X-linked dominant and X-linked recessive disorders. X-linked inheritance, X chromosome. An inactivated X chromosome gets condensed into a small, dense structure in the nucleus, and it is called as a Barr body. Barr bodies are commonly used to determine sex. Generally, X chromosome, males contain an X and a Y chromosome, whereas females contain two X chromosomes. That's why one X chromosome is important to determine the sex. So it forms a Barr body, which is important to determine the sex. X-linked, as related to genetics, refers to characteristics or traits that are influenced by genes on the X chromosome. Humans and most other mammals have two sex chromosomes, X and Y. Females have two X chromosomes in their cells, while males have one X and one Y. In the case of X-linked diseases, it is usually males that are affected because they have a single copy of an X chromosome that carries the disease, causing mutation. Whereas in females, the presence of a second non-mutated copy may cause different milder or no symptoms of the sex-linked disorder. So, as I told, in males they have X and Y chromosome. So since they have only one X chromosome, when the X chromosome is affected, they get mostly affected. Whereas females, since they have two X chromosomes, the other one might not get mutated and make the symptoms milder. X-linked. The way X-linked traits may manifest differently in females and males can be due to more complex phenomena such as X inactivation and exactly how copies of the X chromosomes a person has. A simple rule of thumb for most X-linked conditions is that females generally have a milder version of the condition compared to males. So this explains the same point as the top one. A human male has two sex chromosomes, the X and Y, unlike the other 44 autosomes. Here the X and Y are called as the allosomes. The 44 autosomes, which are the non-sex chromosomes, the X and Y don't carry the same genes and aren't considered homologous pairs. Instead of an X and Y, a human female has two X chromosomes. These X chromosomes do form a bona fide homologous pair. Here, bona fide homologous pair means homologous pairs, that is, they contain the same gene but different alleles. Because sex chromosomes don't always come in homologous pairs, the gene they carry or show unique distinctive patterns of inheritance. Next is a pedigree chart. The actual definition of a pedigree chart is a flow chart or a diagrammatic representation prepared to exhibit the occurrence and appearance of phenotypes of a particular gene or organism along with its ancestors from one generation to next generation. But the simpler definition for a pedigree chart is it is a diagram of family history that uses standardized symbols to show different types of inheritance and diseases passed from one generation to another. The effects of X-linked inheritance, and we are going to learn about how X-linked inheritance happens in dominant and recessive. When coming to dominant inheritance, in males who only have one X chromosome, a mutation in the copy of the gene on a single X chromosome causes the condition. Fathers cannot pass these X-linked dominant conditions to their sons, but all daughters of the affected fathers will be affected with the conditions and can pass on to their children. So in X-linked dominant inheritance, it generally happens because males have only one X chromosome. That is the main reason why they always get affected. In X-linked recessive inheritance, a daughter inherits a single mutated gene on the X chromosome from one of her parents. The X chromosome she inherits from the other parent will usually cancel the effect of the mutation, and she most likely will not have the genetic condition. As we learned from the top slides, why the female doesn't most likely get affected is because she has two X chromosomes. So the other X chromosome mostly mildfies the symptoms. Next, we are going to learn about the difference between X-linked dominant and recessive. X-linked dominant. X-linked dominant disorders are caused by variants in gene on the X chromosome. In males who have only one X chromosome, a variant in the only copy of the gene in each cell causes the disorder. Whereas in females who have two X chromosomes, a variant in one of the two copies of the gene in each cell is sufficient to cause the disorder. Females may experience less severe symptoms of the disorder than males. A characteristic of X-linked inheritance is that fathers cannot pass X-linked traits to their son, and it cannot be confirmed. 100% incidence of affected daughters from an affected father suggests X-linked dominance. Example, Fragile X syndrome. Here in X-linked dominant, both males and females get affected, but the males pass their dominant traits to the females but not to their sons. Whereas in coming to X-linked recessive disorders are also caused by variants in genes on the X chromosome. In males who have only one X chromosome, one altered copy of the gene in each cell is sufficient to cause the condition. In females who have two chromosomes, a variant would have to occur in both copies of the gene to cause the disorder. Because it is unlikely that females will have two altered copies of this gene, males are affected by X-linked recessive disorders much more frequently than females. A characteristic of X-linked inheritance is that fathers cannot pass X-linked traits to their son, and sex linkage cannot be confirmed. 100% incidence of affected sons from an affected mother suggests X-linked recessive. A common and a very famous example is hemophilia, which is also called as the royal blood disease from the Queen Elizabeth family, and Fabry disease. Here in X-linked recessive, all the females are carriers. As you can see the picture, we have an unaffected father and an affected mother in X-linked dominant inheritance, which is equally passed to both their son and a daughter, whereas the other son and other daughter are unaffected. In the other opposite picture of X-linked recessive inheritance, we have an unaffected father and a carrier mother. So the carrier mother passes it to completely as a dominant trait to the son, but she also passes the trait as a carrier to her daughter. So one daughter is a carrier and one is a affected son, and we have one unaffected son and one unaffected daughter. Next, coming to X-linked dominant. Ma'am, can you move the slide?
Okay.
Yeah. Next, coming to X-linked dominant. X-linked dominant inheritance refers to genetic conditions associated with mutations in genes on the X chromosome. A single copy of the mutation is enough to cause the disease in both males and females. Before, we learned that in females, because they only have one X chromosome, it generally mild the symptoms, but whereas in X-linked dominant, if one X is affected, it doesn't mild, it completely affects both the males and the females. This kind of trait affects both males and females equally and does not skip generations. As you saw in the previous picture, both the son and the daughter are equally affected by their unaffected father and affected mother. All affected males have an affected mother, and all affected females have an affected father or mother. All female progeny of an affected male are affected and X-linked dominant with male lethality. Conditions such as Rett syndrome, onychia pigmentation, oral facial digital eye syndrome, focal dermal hypoplasia, and X-linked chondrodysplasia punctata type usually result in early gestational male lethality. X-linked inheritance and examples of X-linked dominant disorders include all the examples like Rett syndrome, the X-linked lissencephaly, and double cortex syndrome, incontinentia pigmenti type 1, characterized by dermatological, ocular, dental, and neurological abnormalities. And the picture shows the X-linked dominant where it has a carrier mother passing it to her son and her daughter. And here another daughter who has married a dominant father. It is passed to both of their daughters. Now my friend is going to continue the next topic of double cortex syndrome.
Hello everyone. From the previous slides, we have learned about X-linked inheritance, difference between X-linked dominant and recessive, pedigree chart, and effects of X-linked inheritance. From the upcoming slides, we are going to learn about X-linked dominant disorders and how the mutation occurs on the genes of X chromosome. So let's start. Double cortex syndrome. Double cortex syndrome is also called as subcortical band heterotopia, which is a rare neuronal migration disorder classically present with seizures and intellectual impairment and is seen almost exclusively in females. Here, neuronal migration disorder refers to abnormal migration of neurons in developing brain and nervous system. As in these patients, there will be a variety of cortical malformations in the brain. Then this syndrome is mostly seen in females, as an affected male passes this trait to daughter, which results in affected females. Most of the females are affected in this syndrome. It is an X-linked genetic disorder with double cortin DCX gene mutation being the causative factor in most of the case, which is related to cortical layer. Cortex is the outermost layer of the brain. It has 2 to 4 millimeter of thickness. Cerebral cortex is mainly responsible for many higher order brain functions such as sensation, perception, and voluntary actions. The overlying brain demonstrates some shallowness of sulci. Incidental cavum septum pellucidum Edwargei is noted. About this point, we will learn at the next slide. Symptoms. Affected individuals typically present with epilepsy and variable degrees of mental retardation. Seizures often start in the first decade of life. They may progress to multiple seizures types and are usually refractory to medication. We can treat this double cortex syndrome by pre-surgical evaluation, including EEG recordings and magnetic resonance imaging MRI. EEG represents electroencephalography, which is a test that measures electrical activity in the brain using small electrodes attached to the scalp. As everyone knows, MRI is high resolution images to see characteristic heterotropic band in the brain. So hence this disorder is also called as subcortical heterotopia. And in this slide at the left side of the picture, it's a normal part of the brain, and at the right side we will see the patients who is affected by double cortex syndrome. As generally, a set of neurons reach the outer layer and properly form at the cortex. At the same time, there are another set of neurons that don't reach the outer layer and form another cortex layer below the outer one. As you see in the right side of the picture, there are many layers formed in the brain. This condition is called as double cortex syndrome, where two layers of cortex are formed. Ma'am, can you show the slide, ma'am?
Yeah, I'm sorry. Someone come in.
This bad. That is the next we will move on to Rett syndrome.
You okay?
Yes, ma'am. Next slide. Thank you. Rett syndrome is a genetic disorder that typically becomes apparent after 6 to 18 months of age, that is 1 to 2 years, and almost exclusively in females. Symptoms include impairments in language and coordination and repetitive movements. Basically, RTT, that is Rett syndrome, is caused due to missense mutation in MFCP2 gene, that is methyl CpG binding protein 2, which results in a significant expression in brain. Those affected difficulty walking and
I'm sorry Nga, but keep silent please. Nga. Em tắt cái mic đi Nga. Okay, I'm sorry. Wait a minute.
So complications of Rett syndrome can include seizures, scoliosis, and sleeping problems. Scoliosis refers to the abnormal lateral curvature of the spine. The severity of this condition is variable. Impairments in language and coordination and repetitive movements, slower growth, and smaller head size. This syndrome can be treated by medications, nutritional support, and speech-language therapy. Next, we will move on to incontinentia pigmenti, which is an X-linked genetic disorder. It is also called as Bloch-Simon syndrome, Balch-Sulzberger disease, and melanoblastosis cutis. Incontinentia pigmenti is an X-linked dominant disorder that affects the skin, nails, teeth, and hair. Symptoms of this disorder is skin redness, warty skin growths. Your warty growths refers to small non-cancerous growths which is seen in the skin when it is affected with one of many human papilloma virus family, and skin decolorations, reduced pigmentation, hair loss, discolorization of nails. We can prevent this disorder by early diagnosis or therapy, which can slow down the progress of the destruction of retina. Next, we will move on to X-linked hypophosphatemia. X-linked hypophosphatemia is an inherited disorder characterized by low levels of phosphate in the blood. As phosphate levels are low, because phosphate is abnormally processed in the kidneys, which causes a loss of phosphate in the urine. This condition is called as phosphaturia and leads to soft, weak bones and rickets. XLH is caused by genetic changes in the PHEX gene. Your PHEX gene stands for phosphate regulating endopeptidase homolog X-linked gene, which is present on the X chromosome, and this inheritance is X-linked dominant. X-linked dominant conditions include X-linked hypophosphatemia, also known as vitamin D resistant rickets. Rickets can be caused by a dietary deficiency of vitamin D. But in vitamin D resistant rickets, the disorder occurs despite adequate dietary intake of vitamin D. As you can see in this picture, there is a normal, ma'am, previous slide.
Okay, thank you. Sorry.
There is a normal picture of human bones, and below in the second picture you can see the condition osteomalacia, which refers to softness of bones due to deficiency or decrease in levels of phosphorus, it leads to rickets or softness of bones, and it becomes weaker. Next slide. Causes of hypophosphatemia. Hypophosphatemia is a serum phosphate concentration less than 2.5 milligram per deciliter. Generally, normal persons should have serum phosphate level of 3.4 to 4.5 milligram per deciliter. Here it is 0.81 millimole per liter, which is deficient, so it leads to condition hypophosphatemia. Causes include alcohol use disorder, burns, starvation, and diuretic use. Clinical features are listed below. In this picture, we can clearly see that how the hypophosphatemia occurs. This picture represents the osteomalacia, rickets, microfractures, and pain. Here, serum FGF3, that is fibroblast growth factor 23, is a phosphate regulating substance which is produced by osteocytes that acts on the kidney. As a result, phosphaturia condition, which is excessive discharge of phosphates in the urine has occurred. So the kidney can't storage much of phosphate, which results in decrease in phosphate reabsorption, and which further leads to decrease in serum phosphate level. This can be done by another method also, by decrease in the 125 dihydroxy vitamin D3, that is also called as calcitrol, which is a hormone which activates vitamin D receptor in the nucleus of the cell. As vitamin D receptors are activated, then only it can absorb phosphorus to our small intestine. As it is in the decreased amount of level, the phosphate absorption of ions also decreases, which leads to a decrease in serum phosphate level. Further, it results in defective mineralization. Mineralization is nothing but calcium phosphate crystals are produced from the osteoblast. As there is a decrease in serum phosphate level, the mineralization process does not occur, which leads to defective mineralization. And this further leads to PHEX gene inactivation. Next slide. Symptoms. The common symptoms of XLH affect the bones and teeth, causing pain, abnormalities, and osteoarthritis. Symptoms in children are rickets, bone pain, fatigue, fractures, delayed growth. And in adults, we can see osteoarthritis, depression, short stature, fatigue, craniostenosis. Craniostenosis is a condition in which the skull gets contracted in adults. Genetics behind hypophosphatemia. It is associated with a mutation in the PHEX gene sequence located on the human X chromosome at location X P22.2 to P22.1. Here X stands for X chromosome, P stands for short arm of the chromosome, and 22.2 refers the exact position of the PHEX gene. This disorder is inherited in an X-linked dominant manner, which is responsible. This means the defective gene responsible for the disorder PHEX is located on the X chromosome. Only one copy is enough to is sufficient to cause this disorder from a parent who has this disorder. So males are normally hemizygous, as they have only one X chromosome and one Y chromosomes. One X chromosome is enough to inherit from parent to the males. As a result, X-linked dominant disorders usually show high expressivity in males than females. Treatment of X-linked hypophosphatemia. Conventional therapy consisted of medications including human growth hormone, calcitrol, and oral phosphate and calcitrol. Unwanted effects of this therapy have included secondary hyperparathyroidism, nephrocalcinosis, kidney stones, and cardiovascular abnormalities. Nephrocalcinosis is a condition in which deposition of calcium in the kidney parenchyma and tubules. And as we all know, hyperparathyroidism is a condition in which the parathyroid hormone is as they increase in parathyroid hormone in the parathyroid gland. X-linked inheritance. As this is X-linked dominant trait, both the daughters and sons of an affected female have a 1 in 2, that is 50% chance of being affected regardless of sex. We can see a clear genetic picture at below of the treatment. To conclude, from these slides of X-linked dominant inheritance, such as double cortex syndrome, Rett syndrome, incontinentia pigmenti, and X-linked hypophosphatemia, we can conclude that in X-linked dominant inheritance, an affected male transmits the trait to his daughters, but not to his son. As a result, the ratio of affected females are more compared to affected males in X-linked dominant disorders. Now, I would like to call upon Ganesh to explain about X-linked recessive disorders. Thank you.
Yeah, thank you.
Good afternoon, ma'am.
Please.
Good afternoon, ma'am.
Yeah.
Now I'm going to tell about X-linked recessive inheritance. Actually, X-linked recessive or sex-linked recessive, genetic condition associated with mutation in genes on the X chromosome. As males have only one X chromosome, if they have a gene alternation on their X chromosome, they will develop the condition. Females rarely show signs of X-linked recessive conditions, as they usually have a second unaltered copy of the gene on their X chromosome. A female who has gene alternation on one of her X chromosome is said to be a carrier for the X-linked recessive condition. Features of X-linked recessive inheritance includes males affected almost exclusively. The gene's mutation can be transmitted from female carriers to sons. Here, what is the genotype of male affected? X, A, Y. Female affected, X, A, X, A. Female not affected, X capital A, X capital A. Male not affected, X capital A, Y. And in X-linked recessive, the carrier will be female. Now, next slide.
Yeah.
Affected males cannot transmit the condition to their sons. Common X-linked recessive disorders, what we study, hemophilia, Duchenne muscular dystrophy, colorblindness, and other some juvenile retinoschisis, nephrogenic diabetes insipidus, cleft palate with ankylosing. But we study about hemophilia, Duchenne muscular dystrophy, colorblindness now. Ma'am, next slide. Hemophilia. This disorder is characterized by uncontrolled bleeding and the inability of the blood to clot properly. Hemophilia, also called as royal diseases, bleeders diseases. Hemophilia is two types normally, hemophilia A and hemophilia B. Hemophilia A is the non-formation of clotting factor 8 and hemophilia B is the non-formation of clotting factor 9. Hemophilia is one of among the many X-linked recessive inherited genetic disorders where the gene causing the disorder of dysfunction is located on the X chromosome. See in this picture, we can see that normal blood vessel, how it turns to hemorrhage, and then clotting. We can compare that in healthy person and hemophilia. Ma'am, next slide. See, the heterozygous female is a carrier for hemophilia, may transmit the disease to sons. The possibility of a female becoming a hemophilic is extremely rare because of mother. Such a female has to be at least carrier and the father should be hemophilic. The mechanism of inheritance of hemophilia. See, in the first picture that made between healthy father and mother carrier, we can see one children will be affected, that is son. Next, two, one healthy son, one healthy daughter, and one carrier. The carrier will be female. Next slide. Color blindness. Color blindness. Color blindness. It is a sex-linked recessive disorder due to defect in either red or green cone of eye, resulting in failure to discriminate between red and green color. It occurs in about 8% of males and only about 0.4% of the females. This is because the genes that lead to red-green color blindness are on the X chromosome. Types of color blindness: deuteranopia, protanopia, tritanopia, and achromatopsia. The symptoms of color blindness that find it too hard to tell difference between red, orange, yellow, brown, and green. And the person with color blindness confuse reds with black. And cause of color blindness, there are several factors which cause a color vision problem in a person. The factor include damage caused to brain or eye or to the nerve cell, genetic disorders, side effects of drugs, use of tobacco and alcohol. Next, what is the treatment for color blindness? Actually, there is no cure for color blindness, but we can use special glasses and contact lenses. Ma'am, previous slide for hemophilia.
This one.
Yes, ma'am. What are the symptoms of hemophilia? Gastrointestinal bleeding, hematuria. Hematuria means blood in urine, severe nosebleeds, hematorosis, bleeding into joint places. What is the diagnosis for hemophilia? Mainly lab tests like platelets counting, prothrombin reaction time. Treatment for hemophilia, injections of missing or non-functional clotting factor. Ma'am, next slide.
This one now.
That's new. Next slide, ma'am. That's
This one.
next. Yes, ma'am. Yes, ma'am. The previous, ma'am. Previous.
This one.
Yes, ma'am. Yes, ma'am. Symptoms of color blindness: rapid eye movement, sensitivity towards the bright light, trouble in seeing colors and brightness of colors. The diagnosis of color blindness: there are certain tests available in diagnosis and to measure color vision deficiency in a patient. Doctors can easily diagnose color blindness using the Istioplate test, but the color blindness won't have proper treatment. Only we can have special glasses and contact lenses. Here we can see pedigree chart that normal man versus carrier woman. One color blinded son and one normal daughter, one normal son and one carrier daughter. Ma'am, next slide. Destiny muscular dystrophy. Destiny muscular dystrophy is a genetic disorder characterized by the progressive loss of muscle. What is the timeline? 2 to 3 years start seeing reduced motor function. 6 to 11 steady decline in strength. 9 to 13 failure to walk. 15 to 25 death most common. Inheritance. Destiny muscular dystrophy is inherited in an X-linked recessive pattern, meaning the gene that carries the Destiny muscular dystrophy causing mutation is on X chromosome. Since males have only one X chromosome, a mutation in gene responsible for Destiny muscular dystrophy is sufficient to cause the condition. What are the symptoms of muscular dystrophy? Frequent falls, difficulty rising from lying or sitting position, trouble running and jumping, large calf muscles, unusual walking, trouble swallowing, heart problems such as arrhythmia and heart failure, learning disabilities, stiff or loose joints, and muscle pain. And what is the treatment for Destiny muscular dystrophy? Treatment steroid medications to obtain muscle strength and some physical exercises to overcome the Destiny muscular dystrophy. Thank you, ma'am.
This one.
Okay, the next.
This is mine, ma'am. This is mine.
Yeah, okay.
I would like to call upon Rokesh to explain about skewed X chromosome inactivation and genetic risks.
Okay, thank you, Susena. Okay, hi guys, I'm Rokesh. I'm going to talk about skewed X chromosome inactivation. Skewed X chromosome inactivation occurs when the X inactivation of one chromosome is favored over the other, leading to an uneven number of cells with each chromosome inactivated. It is usually defined as one allele being found on the active X chromosome is over 75% of cells, and the extreme skewing is even over 90% of cells have inactivated the same X chromosome. Anyone of you understand from this? Nothing. Okay, let me try this from my own words. Skewed X chromosome inactivation. Everyone listen, guys, please. Everyone has
two sex chromosomes. Girls are X and X and boys are X and Y. Almost all of the information is on the X chromosome. So if girls have twice as many X's, they have twice as much information. So our cells deals with this by a process called X inactivation. Okay, how does the X chromosome inactivation occurs? If a zygote starts, there is a cell, right? If a female zygote, there is an X and X cell will be there. On the two X chromosomes, the cell will divide. All of the cells, new cells, have two X's. Two X's. Soon she is a ball of cells, and each cells have two X chromosome. At this point, each cell decides to inactivate one of its X's chromosome. Understand that? The X that turns off is random, and each cell can make a different choice. But each cell choice is permanent. If there's a 2X, right? If first X turn off, if the cell divides, another the child of the previous cell is first X of the first chromosome will be turned off. Okay. After each chooses, all of its future copies have the same X turned off. Therefore, girls are called mosaics of X's. In girls, this is called squid X inactivations. This doesn't happen in boys. Do you know why this is not occur in boys? Any one of you? Ma'am, can I interact with them, ma'am?
Yeah. Yeah,
Any one
you
of
can. You can do it.
Yeah, thank you, ma'am. Okay, let me ask one question. Any one of you, sir? Sir, you can also join with this conversation, sir. Like, I'm going to ask one question, sir.
This one slide. Or next, this one.
Okay, ma'am. Okay. Shall I continue, ma'am? Hello? Okay. This doesn't happen in boys because they only have one X chromosome, because boys are X and Y, right? Because boys have only one X chromosome. The good example is for the squided X inactivation is Kelly-cat. This is a normal process. It evens out information between boys and girls. Okay, diseases by squid inactivation is X chromosome aneuploidy. Results in disease phenotypes in human. Klinefelter syndrome. And Klinefelter syndrome is where boys and men are born with an extra X chromosome. That is defined on the slide. Triple X syndrome. Delayed learning, delayed speech, language, motor skills, learning disabilities, reading, understanding, whatever. And ma'am, can you show the next slide, ma'am?
Okay.
Next, ma'am.
Next.
I am genetic risks. The X chromosome from a male is transmitted to daughters, and the Y chromosome is transferred to sons. If an affected male has kids with a healthy female, the male offsprings of that male will not get affected, but all of his female offsprings will be the carriers. If a carrier female has kids with a healthy male, each male offspring has a 50% chance and being affected, and female offspring have a 50% chance of being a carrier. Next slide. Conclusion. Okay, this is the end of our presentation. If you are a human being, most of our chromosomes come in homologous pairs. The two chromosomes of a homologous pair contain the same basic informations. That is the same genes in the same order, but may carry different versions of those genes. Are all of your chromosome organized in homologous pairs? The answer depends on whether you are male or female. X-linked recessive inheritance is a mode of inheritance in which a mutation in a gene on the X chromosome causes the phenotype to always express in males and in females who are homozygous for the gene mutation. See zygosity. Sex-linked diseases are passed down through families through one of the X or Y chromosomes. X and Y sex chromosomes dominant inheritance occurs when an abnormal gene from one parent causes diseases, even though the matching gene from the other parent is normal. Thank you, guys. Thank you, ma'am.
Yeah, thank you so much.
Thank you, ma'am.
And so we have one more group. Last one is cytoplasmic inheritance. We'll have presentation by group 1. Okay, you are ready? Who will have this presentation? Let's see. Hasita Saloni. Group 1, are you ready?
Yes, ma'am.
Yes, ma'am.
Okay.
Yes, ma'am.
Let's
Yes,
see.
ma'am.
Wait for me. Spoken language here, English India, and subtitle language here. Let's see. Maybe still continue with Vietnamese. So check. Then leave. Okay. Okay, here we go.
Ma'am, next slide.
Okay, next slide. Okay.
One more. Next.
One more.
Good morning, everyone. Now we are here to speak upon, good afternoon, sorry, upon the topic inheritance. Basically, what is inheritance is the transmission of genetic characters from one generation to the other. There are 6 ways through which the data can be, the genetic data can be transferred. Autosomal recessive, multifactorial transfer, X-linked recessive, autosomal dominant, cytoplasmic inheritance, and S-linked dominant. Next slide. Ma'am, next slide, please.
This one?
Next slide. What? Yes, ma'am. Correct. No. Back. Cytoplasmic inheritance. Yes. What is cytoplasmic inheritance? Basically, there are 2 types of inheritance. One is nuclear inheritance, and the another is cytoplasmic inheritance. In nuclear inheritance, the nucleus provides the genetic material to be transferred to the next generation. But in cytoplasmic inheritance, the genetic material is provided from any of the organelle which is located inside the cytoplasm. For example, either mitochondria or plastid. If we take the example of human beings or mammals, the mitochondrial DNA is transferred from mother to the offsprings. And plants, the plastidial DNA is transferred from one generation to another, which carries the information of the color of the plant and many other phenotypes. Next slide. Others?
Another one?
No, ma'am. Maternal inheritance.
Good afternoon, ma'am.
Yeah.
Maternal inheritance. In generally, the character of only one of the 2 parents is transmitted to the progeny. The transfer of mitochondrial DNA is from mother to the offspring, ma'am. Hence, they are referred as extranuclear, extrachromosomal maternal inheritance or uniparental inheritance. In most animals, the parental mitochondria enter the oocyst cytoplasm after the fertilization. The mitochondrial DNA is never transmitted to the offspring. This pattern of mitochondrial DNA inheritance is known as maternal inheritance. Example, shell coiling in snail. In snail, the shell is spirally coiled. Okay, the snail exhibit 2 type of coil. The shell coil to the right is dextral and the left is sinistral. This gene of dextral is a dominant and the recessive is a small d. Next slide, ma'am.
Yeah, that's okay.
Hello. Yes. Hello. So I I'm about to. Can you mute it? The cytoplasmic inheritance of color in plant by plastid is called the plastid inheritance. It was first described by Carl Corns in 1908, and it is best described in the 4 o'clock plants, that is the Mirabilis salapa. So in the Mirabilis salapa, it has like 3 types of branches. The green leaves or branches, the pale leaves, and the variegated leaves. So the green leaves has plastic chloroplast, and the pale leaves has leucoplast as their plastic, and the variegated branch has chloroplast, leucoplast, and chromoplast. So that is why they are producing a particular colored branch. So in this plant, if the green plant produces an egg, the female gamete that is then the egg, uh, the gamete will have nuclear material, and in its cytoplasm there will be a chloroplast. And if it is fertilized by the male gamete coming from any other branch, the male gamete is not going to bring any cytoplasm. So when the zygote is produced, the color of the branches and the leaves will be based on what comes from the cytoplasm of the female egg. So so the color of the zygote will be green in color because the mother has the chloroplast. And so in case of variegated branch, the female gamete has 3 types of plastids. All of the plastids, that is the chloroplast, leucoplast, and chromoplast. So here the offsprings are going to be of 3 types, that is green, pale, and variegated. And this means that when the cytoplasm has 3 types of plastids, whichever the plastid goes in maximum number, the color of the branch will be seen in the offspring. So from this, we can understand that the color of the branch in the progeny is decided by the plastid which is present in the cytoplasm, which comes only from the egg or the female gamete. So that is why it is under cytoplasmic inheritance. Thank you.
Okay, sorry. Okay, who's next?
Next slide.
Okay,
Yeah,
not yet. Okay, wait. This one?
yes, ma'am.
Good afternoon, ma'am.
Others?
Yeah, good afternoon, ma'am. Myself, Father. So, mitochondrial inheritance. So, mitochondria, we all know mitochondria is the powerhouse of the cell or the energy source. And the one thing is, mitochondria have its own DNA. So, that is the mitochondrial DNA. The DNA of the mitochondria comprises totally 1% of cellular DNA. And in this case, mitochondrial inheritance, the most interesting thing is the DNA of the mitochondria is only inherited from mother and not from father. So you guys can tell to your mother that she gave me something that dad never could. So in this case, mitochondrial inheritance. By this also we can refer to just look at the diagram. We can see that sperm also have mitochondria and ovum also have mitochondria. But in the case of zygote, there is also mitochondria which is totally contributed by mother. So from this we can understand it does not obey Mendelian's law of inheritance. That is, mitochondrial inheritance is non-Mendelian, meaning it does not conform to the Mendelian laws of inheritance. And at last, the mutations in the mitochondrial DNA could lead to some of the diseases that's called mitochondrial diseases. And we have just included one of the most important disease in this QR code, and you guys can just scan it and go through it. It's just for reference and not a big deal. So the disease included in this is the Leber's hereditary optic neuropathy. So you guys can just scan this QR code and just go through it. And that's all for my topic. And I'm shifting my position to Richard, and he will take the rest. Thank you.
Ma'am, next. Yes, ma'am.
Next slide.
I'll be explaining the significance of cytoplasmic inheritance. Significance means like the importance of cytoplasmic inheritance. Cytoplasmic inheritance help us to determine the importance of the cytoplasmic organelles, organelles like those ribosomes, plastids, in passing on the characters in several organisms. The second point is cytoplasmic inheritance help us to map the chloroplast genome and mitochondrial genome in many species, such as humans, yeast, maize, etc. The third and fourth point, it is same with the first point, so I will add a different points. Cytoplasmic male sterility has developed in crops, and because of this, the plants like maize, cotton, palm, etc., it has helped in production of more hybrid seeds. So there is more production of these plants, and the cytoplasmic mutation help us to produce new variations of generations. From cytoplasmic inheritance, these mutations are observed in chloroplast and mitochondrial DNA. From cytoplasmic inheritance, we can produce new variations of characteristics. So these are some of the significance of cytoplasmic inheritance. Now, I pass my position to Ajay.
Good evening, everybody. My name is Ajay, and I'm going to explain the pedigree analysis of cytoplasmic inheritance. What is pedigree analysis? The inheritance pattern of traits, which can be determined by the analysis of family trees, are known as the pedigree analysis. As you guys can see in the diagram, the affected children, mother and father, are denoted in the red color. You can notice the genes are passed through the mother and not through the father. It's because the cytoplasm of the zygote is provided by the ovum and not by the sperm. Ma'am, is it okay if you can share a screen?
Yeah, you can share screen. Hey, Ajay.
Yes, ma'am.
Yeah, okay, yeah.
As you guys can look in the diagram over here, the circle denotes a female and the square denotes a male. The circle is where the female is infected with the mitochondrial inheritance, and you can see that the gene is passed on to the 4 of the family members. But if you look down to the 3rd generation, you can see clearly that only the females who are infected have been passed those genes to the both males and females, whereas the males who have been infected have not been passed on. As I said on my previous sentences, that the cytoplasm will be passed only by the ovum and not by the sperm. That's it, ma'am. Thank you.
Yeah, thank you, Ajay.
Hello, everyone. I'm here to explain about the introduction to sex chromosomes X and Y. A human male.
Aurora.
One slide is not visible.
And you want me to share screen again?
Yes,
ma'am.
Okay. Here. Yeah, can you see?
Introduction to sex
Yes, ma'am.
X and Y. A human male has 2 sex chromosomes, the X and the Y. Unlike the 44 autosomes, these are the non-sex chromosomes, and the X and Y, these are the sex chromosomes. These X and Y sex chromosomes doesn't carry the same genes and aren't considered homologous. Instead of an X and a Y, a human female has 2 X chromosomes. The X chromosome contains genes which develop female characters, and the Y chromosome contains genes which develops male characters. And let's know about genes on Y chromosome. Genes of male reproductive parts. It also contains genes of baldness, genes of male hormones, and genes of hypertrichosis. Thank you.
You finish?
Hello, ma'am. Next slide.
Okay.
Previous slide, ma'am.
Previous, this one.
Good evening, everyone. Today I'll explain the topic on genes on Y chromosomes. Genes of male reproductive parts. The SRY gene provides instructions for making a protein called the sex-determined region Y protein. This protein is involved in male typical sex development, which usually follows a certain pattern based on an individual's chromosomes. The next one is genes of baldness. The balding gene is explained as when hair loss is caused by genetics. It occurs on a predictable pattern, often referred to as male pattern baldness or female pattern baldness. The next one is genes of male hormones. The AR gene provides instruction for making a protein called an androgen receptor. Androgens are hormones such as testosterone that are important for normal male sexual development before birth and during puberty. The next one is genes of hypertrichosis. Congenital generalized hypertrichosis terminalis is thought to be caused by genetic changes on chromosome 17, resulting in addition or removal of millions of nucleotides. The gene MAP2K6 may be a factor contributing to this condition. Thank you. I'll hand over to Divya Darshini.
Good evening, ma'am.
Ma'am, next slide.
This one, right?
Ma'am, correct slide. It's
This
correct.
one?
No, back.
This one?
No, ma'am, why? Yeah, correct.
Okay.
The topic is Y-linked inheritance. The inheritance of a gene on the Y chromosome is called as a Y-linked inheritance. Both male and female have X chromosome, but in male alone we can see the Y chromosomes. So we can see the Y-linked genes can transmit from the father to sons. The Y chromosome is generally very smaller than the X chromosome, so it can carries only fewer genes. Beside the genes for sperm development, specific hormonal outputs and other traits characterizing male, the Y chromosome also carries the genes other than for sex determinations. The Y chromosome is quite smaller and comprises a few genes. And the majority of the genes are linked to the male infertility and spermatogenesis and are being identified as the long arm of the Y chromosome. And next slide, ma'am.
So good evening, ma'am. I'm here to explain about the Y-linked disorders. So the first disorder is hypertrichosis. So as the name says, hypertrichosis is nothing but the excessive amount of hair growth, occurrence of too much of hair growth all over the body. And it can also be localized, that is the hair growth over a certain body part. So and therefore it is called as localized hypertrichosis. And the next disorder is webbed toes. Webbing of toes or fingers in man is mainly due to a local arrest of development, leading to retention of the normal embryonic webbing. This form of digital fusion includes only the skin, and the skeleton remains unaffected. The extensor tendons of the toes might sometimes be fused. The extensor tendons are nothing but the small tendons that are located beneath the skin. Porcupine man. Ichthyosis hysteris or the porcupine. Ma'am, next slide.
Sorry, wait for me.
So porcupine man. Ichthyosis hystris or the porcupine man refers to a group of rare skin disorders in the Ichthyosis family of skin disorders that is generally characterized by a massive hyperkeratosis, along with the appearance like spiny scales. This is mainly a rare condition of autosomal dominant inheritance. However, few sporadic cases do occur. And the last disorder is infertility in male, in which the Y chromosome infertility is a condition that affects the production of sperm and causes male infertility, which means it is difficult or impossible for affected men to father children.
Who's next? Hey, can you hear me? Yeah.
Hello.
Yeah, hello.
Pedigree analysis of Y-linked. When completing this pedigree with Y-linked inheritance, the trait is carried on the Y chromosomes and it is transmitted from father to son only. When writing genotype, we still use X and Y as a symbol for the chromosome passed on from the previous generation. But only the Y chromosome will have an allele. For example, all the females will have the genotype XX, no alleles. Male with the trait will have the genotype XYR, and male without the trait will have the genotype XY small r. With the allele on the Y, the alleles are not dominant over one another, so the R and the R were just randomly assigned as a symbol to represent each allele. Patterns of Y-linked inheritance. After filling in the genotypes for the individuals in the several family tree that exhibit this mode of inheritance, some pattern that can be noticed are trait expression and transmission is only in the males, the individuals with the Y chromosome. If a male has a trait, so should his father and parental grandfather, as well as his son and their son. It follows the inheritance of the Y chromosome. Thank you.
Yeah, thank you. And so the end of your presentation now.
Yes, ma'am.
Yeah. And so we have just finished. And so do you have any question?
No, ma'am.
No. And so thank you so much for your attending today and your night presentation. You have prepared a lot of time, huh?
Thank you, ma'am.
Yeah. Okay. And so because this week we have no class anymore on our room, and so can we continue to have the online class like this?
Okay, ma'am. That's good.
Okay. Okay. And so I'll see you on Wednesday morning.
Thank you, ma'am. Thanks a lot.
Okay. Thank you so much. Okay. See you later.
Yeah.
Yeah, ma'am.
Thank you, ma'am.
Okay. Okay. You leave the call. I will have Mr. Our department. Okay. Thank you so much.
Thank you.
Thank you, ma'am.